All right, team, I hope you're well rested. We're about to hit Chapter 9, which is all about cellular respiration and a slight variation of that, which is called fermentation. This is a big, heavy chapter. If you look through the chapter in the textbook, and if you've looked at this type of material before about cellular respiration, you're probably a little bit worried about this chapter, and I wouldn't say that you shouldn't be worried at all. But I want us to keep in mind the big picture. I don't want us to get bogged down into the tiny little details where you can easily get lost. We want to try to learn as much as possible about those details, but we want to think about big picture. So we're going to step through this bit by bit, and we're going to try to use analogies and everyday language and clear explanations to try to talk about exactly what's going on and why this process is occurring. And hopefully at the end of this, you'll have a little bit better understanding and you'll feel a bit more comfortable about this complex topic. Our overview for the chapter says that life is work, and that is certainly true. For you to have living things which are highly organized against the entropy of the universe, that requires energy. That requires energy from outside sources. So you have animals, such as this lovely little chimpanzee here, animals that eat other living creatures. They either eat other animals or they eat producers, such as plants. And then, of course, you have the plants themselves that have the ability to capture the sun's energy and create their own food via photosynthesis. We'll talk about that in the next chapter. But most living creatures have to ingest something or someone else, plants or other organisms. Generally speaking, in an ecosystem, energy flows into the ecosystem as sunlight, as solar energy and light energy, and it leaves the system as heat. Photosynthesis, which we'll talk about in the next chapter, generates oxygen and organic molecules, which are then used in cellular respiration. So it's the plants that always serve as the base of any food chain. They're the only ones that can take light energy from the sun and form that into complex chemical energy that's stored in molecules that are then used by everyone else. Cells use chemical energy stored in these organic molecules to regenerate and generate ATP, adenosine triphosphate, which is the currency of energy for living creatures, which is the molecule that living cells use to power themselves. So here's a quick model of how the two are interrelated, photosynthesis and cellular respiration. Now we're only going to be focusing on cellular respiration in this chapter, and in the next chapter we'll talk about photosynthesis. So light energy comes into the system, and via photosynthesis that light energy can be transformed into stored chemical energy in organic molecules, plus it gives off oxygen. Those organic molecules can then be used in cellular respiration to create ATP, which can be used to do work. Byproducts of that are carbon dioxide and H2O, which can then return back to the system as part of photosynthesis and be recycled again. So we're going to be looking at primarily eukaryotic creatures, but keep in mind prokaryotic creatures also do cellular respiration. They don't have all the little organelles. They don't have the mitochondria, which is primarily where we're going to be. It's where cellular respiration takes place. But they do cellular respiration within their cytoplasm, and I'll mention that. So we're going to focus on the mitochondria of eukaryotic cells and cellular respiration and how we use organic molecules to create energy and ATP. And then, of course, a lot of that energy gets lost. When I say lost, remember, I just mean not used in the way that we might want it, and goes back into the environment as heat energy. So our very first concept, concept 9.1, says catabolic pathways yield energy by oxidizing organic fuels. Some of those words we know, some of them maybe not. Catabolic, remember, is the breaking down and releasing the energy. So when you talk about a catabolic process, you're talking about breaking it down and releasing the energy. So digestion is a good example of a catabolic process. And it says it yields energy. That's good. That's what we're looking for. By oxidizing organic fuels. This word here, oxidizing, may be one that you're familiar with or perhaps not. But by the end of the chapter, we will understand what oxidizing means. As with most subjects in biology, there's not one simple, clear path. There's multiple kind of processes and steps that are involved here. So that's why this gets so confusing, is because of all the variety of steps, but we're going to try to go through it in a linear fashion, step by step, and try to explain everything. And again, remember, catabolic means the breaking down of complex organic molecules to release that stored energy. We're going to break this catabolic pathway kind of into two major sections. One of those is kind of a method that is not the primary method of, say, humans or dogs or creatures like that. It's called fermentation. Fermentation is the breakdown of organic molecules without using oxygen. And so there are some creatures or in some environments that don't have the necessary oxygen in the environment, so they use a different method called fermentation. And we'll talk about that a little bit toward the end of the chapter. The main one we're going to look at, though, is called aerobic respiration. Aerobic respiration is going to be the breakdown of organic molecules, but it requires oxygen, and it yields ATP, which are the energy molecules that we're trying to create. Now, there is another type that is related to respiration called anaerobic respiration, and we're going to find out that it's very similar to aerobic respiration, but anaerobic means that they use something other than oxygen. So this is a type of respiration that can take place in an environment that has low or no oxygen, and it's going to be very similar, but it uses something other than oxygen as its as the basically the energy source or the pull for the electrons. We'll find out. We're going to focus mostly on eukaryotic organisms like dogs and donkeys and humans. Most eukaryotic and a lot of the prokaryotic organisms also perform aerobic respiration, so that's going to be our main focus, but not all of them. The basic pathway for any of these exergonic, which is, remember, releasing energy pathways, the basic pathway is an organic molecule plus oxygen yields carbon dioxide, water, and energy. So in aerobic respiration, we're taking organic molecules, combining them in a process with oxygen, and from that process we're going to get carbon dioxide, water, and energy. Fermentation is slightly different because it doesn't use oxygen. Anaerobic respiration, slightly different also, uses a lot of the same steps but uses something other than oxygen. When you hear the phrase cellular respiration, that could mean both aerobic and anaerobic, but oftentimes when people say cellular respiration, they're talking about eukaryotic cells and aerobic respiration. So when you see this phrase cellular respiration, you're probably okay in assuming that they're talking about the oxygen using form, aerobic respiration. And although we're going to look at glucose, glucose is going to be our model, and glucose is a carbohydrate, of course. Your body can use carbohydrates, fats, proteins. All of these can be consumed as fuel and used in different parts of this pathway. We're going to choose to use glucose, which is a carbohydrate, a sugar, because that's just a helpful model to use. But I don't want you to think that glucose is the only organic molecule that can be used for cellular respiration. It's not. Your body uses proteins from the hamburger that you eat, and it uses fats from the butter that you put on your toast. All of those materials can be consumed by the body in some way as part of this pathway. So what exactly is going on inside those cells? When you eat a hamburger for lunch, exactly what is occurring after that hamburger gets digested and broken down and that material goes to the cells? Well, we have a couple of chemical reactions that are occurring. And so remember, when we talk about chemical reactions, we're talking about the movement and transfer of those valence electrons. Remember those outer shell valence electrons? Those are what's involved in chemical reactions. And when you move those electrons around or you transfer them from one atom to another, you have changes in the amount of energy. Sometimes that requires energy to go in, sometimes that releases energy when you combine them in different ways. So this energy that's being released is because of chemical reactions, and it's because of the changes in the valence electrons of these atoms. These reactions, we're going to find out, are called oxidation and reduction. We'll get to that in just a moment. But ultimately, what are we using this energy for? Well, the human body and the oak tree and the dog, all of those cells of those eukaryotic organisms, of these living creatures, are using that energy ultimately to create ATP, which is the energy molecule that we use, that our cells use to store and then use that energy. So ATP is kind of like the battery, like a AAA battery that the cells are creating so that then they can use that energy as they need it. So unfortunately, it's not an easy process. It takes a lot of steps to get there, to get energy out of that hamburger and into your cells as ATP. That's a kind of a complicated process, but we're going to step through it bit by bit. To understand some of this, we need to know some basic chemistry. So I don't want to scare you, but we're back to a little bit of basic chemistry. So these chemical reactions, remember, they're moving around and transferring these valence electrons. Specifically, these reactions are called oxidation reduction reactions. So we kind of have a shorthand name for that called redox. So redox reactions, perhaps you've heard of that before. These are oxidation reduction reactions. So let's take a look at what these words mean. In oxidation, oxidation means that the substance loses electrons. So that when we say a substance is oxidized, we mean it has lost electrons. So oxidation is losing electrons. The word reduction means it has gained electrons. And this, I know, is very confusing because when I see the word reduction, I think reduce. So that makes no sense to say that it gains electrons. So there's another way to think about this. In reduction, perhaps a better way to think about it is that it loses positive charge. The amount of positive charge is reduced. So in reduction, a substance is gaining electrons. Or another way of thinking about it, if it helps you, is it's reducing or losing positive charge. Let's take a look at a simple example and perhaps we can kind of figure out what's going on. So remember, this is going to be an ionic bond because they're transferring electrons. But we're talking about sodium, Na here, and chlorine. When we put those two together, they create NaCl, which is salt, table salt. So let's take a look at this reaction. We have sodium, chlorine. Sodium becomes oxidized. Remember, it has that one valence electron that it really wants to get rid of. So it loses an electron, which means it becomes oxidized. Chlorine, remember, has 7 valence electrons, would really like to gain another. So it is reduced. It gains an electron. And again, the problem usually becomes right here where students look at this word reduced, and the fact that that means it gains an electron can be very confusing. So another way maybe to think about it is reduction means reduction in positive charge. Or another way that I think about it is it's becoming more negative. So if you think about like a math problem, if something becomes more negative, that is reduction. It is losing. If you had $5 and now you have $2, things are going more negative. Your amount is being reduced. So that's two different ways you can possibly think about it that will help you out. So reduction either means a reduction in positive charge or it's becoming more negative. It is being reduced. So in this case, sodium becomes oxidized, meaning it lost an electron, it's more positive. The chlorine gains an electron, which means it's more negative, and we call that reduced. Here's another example. This is just using the letters X and Y. They don't represent any particular element. So here we have an ion. This one's already negatively charged. It has an extra electron. And then we have the Y. In this combination, the X becomes oxidized. It loses an electron and becomes more positive. The Y gains that electron and it becomes reduced. It has been reduced in positive charge. It has become more negative. So remember again, reduction can be thought of as reduction in positive charge, or it's becoming more negative, which means it has gained negative charge. Now, in any situation where electrons are going from one atom to another, you can look at it from the perspective of the atom that gains the electron, or you can look at it from the perspective of the atom that loses the electron. So you can kind of look at it as from the perspective of the giver or the receiver. So let's talk about it as far as the perspective of the giver, the electron donor. This would be the atom that gives up the electron or loses the electron. It's called the reducing agent because it is reducing the other atom that it's giving the electron to. The electron receptor, this would be the receiver, is called the oxidizing agent because it's the one that is taking on the electron. It's doing the oxidizing. In other words, it's making the other material more positive. So we can talk about reducing agents. They are the atoms that do the reducing to the other atoms. And we can talk about the oxidizing agents. This is the atom that oxidizes the other atom. So that pretty much makes sense. If we're talking about like NaCl, we're talking about an ionic bond where electrons are given and received. But what if we're talking about a covalent bond? A covalent bond where the electrons are shared. They're not necessarily completely given or received. Well, some redox reactions do not necessarily transfer the electrons fully, like in an ionic bond, but they do shift the sharing. So if we shift the sharing so that the sharing is more uneven, that can also be considered a redox reaction. And a good example of this is when methane reacts with oxygen. Methane is a gas that can burn. Methane is the gas that is in your farts. Methane is the gas that you may have even heard about where they talk about how cows produce so much methane that it affects global warming, that type of thing. So methane is a flammable gas that when combined with oxygen, if the energy is high enough, the heat is high enough, it will burn. And burning is an oxidation reaction. But this is a sharing of electrons. It's a covalent connection. So let's look at the redox reaction when electrons are shared. So let's kind of take a look at this reaction and go through it very slowly so we can understand. So we have methane, CH4 is a methane molecule, and then we have oxygen. We have two oxygen molecules here. We combine those together with enough energy to get the whole thing going. And what comes out of this is carbon dioxide, a fair amount of energy, and some water. So let's find out, how is this a redox reaction? What is being reduced and what is being oxidized? So in this case, we don't have electrons necessarily being completely given away and received, but we have a difference in the sharing of electrons. So here the sharing shifts away from carbon and towards oxygen. Let's take a look and see how that occurs. So first let's take a look at the carbon. So here we have a carbon, and it's bonded to 4 hydrogens. And if you'll remember from a couple of chapters ago, we talked about how this would be a pretty equal sharing. So the electrons that these guys are shared fairly evenly. But in this reaction, this carbon becomes part of carbon dioxide. And if you look at this molecule, carbon dioxide, this is not an even sharing situation. Remember, oxygen is very electronegative. It will share, but it's kind of a greedy sharer. And notice how these electrons are shifted toward the oxygen. So they're shifted more toward the oxygen than the carbon. So we are sharing, but we're not necessarily sharing equally. Now you'll notice that here, everything was shared equally. Here it's not. So carbon has lost a bit of the electrical charge, you could say, the negative charge. So it didn't necessarily actually lose electrons. If you count the electrons, carbon still has the same. But as far as the sharing is concerned, it's no longer getting its fair share. So we would say it has become oxidized. It lost negative charge. Or in this case, it lost a bit of the negativity because it's shifted more toward oxygen. Let's take a look at the oxygen. Between two oxygen molecules, the electrons are evenly shared. So you'll notice they're kind of evenly shared. Now, this oxygen becomes reduced in part of the H2O, water. We know that's an uneven sharing because we know water is a polar molecule, and the oxygen is getting more than its fair share of the electron negativity. So it has become more negative. It has become reduced. It has reduced in the amount of positive because it has become more negative, so it is reduced. So in this case, we have a sharing of electrons, and it's really just a shift in the sharing. The carbon has lost some of its electronegativity as far as the negative charge, and the oxygen has gained. So this is still a redox reaction, but it's a redox reaction as part of a covalent bond reaction. So let's talk about food molecules, organic fuel molecules, and oxygen. You guys eat food, and that goes through your digestive system, and those molecules are broken down small enough to go out to the cells. You breathe in oxygen through your lungs. That oxygen is carried by the blood, again, to the cells. So they have the ingredients that they need. During cellular respiration, the fuel, we're going to model ourselves off glucose, the fuel is oxidized, and then the oxygen, the O2, is reduced. So remember, that means that the electrons are moving away from the fuel and moving toward the oxygen. The fuel is becoming more positive. It's oxidized. It's losing electrons. And the O2 is gaining electrons or becoming more negative. It is reduced. And oxygen is very greedy. Oxygen is very electronegative. It's one of the most potent of all oxidizing agents. And that's why we call it oxidation, because oxygen is the main model that we look at because it's so good at doing this job, at pulling electrons toward it because it's so electronegative. So how does this release energy? Well, here's the key right here. Electrons lose potential energy when they're moving toward a more electronegative atom. This is kind of like moving down a hill or falling down a set of stairs. Remember we said when electrons fall to a lower, more stable energy level, they give off that extra energy, that potential energy that they had, it goes somewhere else. It can be used for something else. So what happens is, as electrons move from the fuel source, say glucose in this case, toward the oxygen, that's a more stable, lower energy state. So where did that energy go that the electrons had when they were as part of the glucose molecule? Well, that energy can be used for other purposes. For the most part, we're going to be talking about how that energy is used to then create ATP molecules. So electrons lose potential energy when they move toward a more electronegative atom. And that's like a ball rolling down a hill or falling down a set of stairs. That potential energy that they are giving up or losing, we call it losing, but remember it doesn't disappear. That potential energy can be used to do work, and in this case that usually means creating ATP. So let's really pause right here because this is a big idea, and if you can kind of understand what's going on here, you kind of understand the whole process, and I'm talking from a big idea perspective. So again, let's do this closely and carefully. So let's take a look. We have a molecule of glucose, and then we have the oxygen molecules that we're combining it with, and then it's going to yield carbon dioxide, which you know you breathe that out as a byproduct, water, which you also breathe out with every breath, and energy, which is what we need. We need that energy. We're going to use that energy to create ATP. And then those ATP molecules can be broken down, and that energy can be released so that you can ride your bike and go to school and drive your car and dance with your girlfriend or boyfriend and do whatever it is you want to do. In this case, the glucose is becoming oxidized and the oxygen is becoming reduced. So the oxygen is taking on more of the negative charge, or it is taking on a greater amount of the negative charge, and the carbon, the glucose is being oxidized. It's becoming more positive. So let's take a look and see what this means. A good example of cellular respiration using our model glucose. The electrons of the hydrogen atoms moving from glucose to oxygen fall to a lower, more stable energy state, releasing energy ultimately used to create ATP. So these hydrogen atoms, as they move from glucose to the oxygen, they fall. They carry, you have protons, of course, but their associated electrons fall in energy state. They fall to a lower, more stable energy state because oxygen is more electronegative. It's basically easier to be close to oxygen. And so that energy that is lost, we keep saying the word lost, but remember it's available, can be used to do work, and in this case create ATP. So a good question is, if that's the natural state of things, if it's easier for the hydrogen and the electrons of the hydrogen protons to fall and be closer to the oxygen, that's like going downhill. That's like rolling a ball downhill or water moving downhill. That's the natural state. That doesn't require energy input. So the question is, if that's a spontaneous reaction, remember that phrase, spontaneous, if that's a spontaneous reaction, how come it doesn't happen all the time? How come glucose isn't just basically bursting into flames? It's not being oxidized at all times within our body. How come chemical reactions are just not running crazy? Well, if you'll remember back a chapter or two ago, you probably already know the answer to this. The reason that these chemical reactions are not just going crazy inside your body, inside the cells, happening all the time, is because remember, every chemical reaction has something called the energy of activation. There has to be some little input. Remember that first hill that you have to kind of overcome, a speed bump you have to overcome before the spontaneous reaction can occur and move forward on its own? Well, that is what allows or keeps these reactions from just going on all the time. That energy, usually in the form of heat, can be added. If we added enough energy to glucose in the presence of oxygen by, say, adding heat, by putting a flame to it or something like that, well, then you'll see this reaction occur spontaneously. The glucose or the other material, it could be, say, cellulose, that will happen. So when you start a fire, a campfire, you're taking cellulose, which is a organic molecule, and you're adding heat, say in the form of a flame from a match, in the presence of oxygen. Suddenly it begins to burn. That's an oxidation reaction, and then that occurs spontaneously. And once that occurs, you get carbon dioxide, you get water vapor. If you didn't know this, fires create water vapor, and you get a lot of energy in the form of heat and light. This spontaneous reaction doesn't occur all the time inside your body because of that energy of activation. So what does your body do? Well, your body uses enzymes to lower the energy of activation to allow this reaction to go on. So if you have, say, a stick of wood at 98.6 degrees, it will not burst into flames. You have to add more heat before that will happen. Well, within your body, this glucose will not oxidize at 98.6. That's not enough energy, not hot enough. So what does your body do? Well, remember, it uses enzymes to lower that energy of activation so that this process can then occur at 98.6 degrees. And your body can regulate how quickly and how often this occurs by regulating those enzymes. Ah, amazing, cool, big idea. So think about this particular page or think about this particular slide and try to wrap your mind around what we're saying here, because this is kind of a large idea about cellular respiration. All right, so you wrapped your mind around that first big idea. Let's go ahead and hit a second big idea. The second big idea is that there's a reason that cellular respiration involves so many steps. We can oxidize our fuel and get all of our energy out at one time. But that's more like an explosion. And we don't want an explosion. We don't need, our cells can't use a sudden release of energy all at one time. So instead, there are multiple steps. And these multiple steps keep the energy release in manageable amounts and forms. So a good analogy is you have a gas tank full of gas in your car. Well, you have a couple of options. You can blow it up. So blowing up your tank of gas will create a huge release of energy all at one time. And it will probably propel your car forward for a certain distance. But you won't go nearly as far as if you had a controlled release of energy. If you had an engine and you burned that gasoline small amounts at a time to release that energy in small amounts at a time to propel you and down the road. So with a tank of gas, you could probably go 300 miles at a more steady, controlled rate, or you could release all that energy in one massive, giant explosion, and that might propel you 2 miles up and down the road, but that would be it. And it would probably do some damage as well, obviously. So there are multiple steps in the cellular respiration process to control the release of energy so that it can be used in the way that the cells need to use it. Every step in cellular respiration requires an enzyme to move on to the next step, and every step moves electrons from glucose. It also moves protons, so we're talking about hydrogen atoms, but it's moving the hydrogen atoms, including their electrons, from the glucose down the chain toward oxygen. So we're going to be moving electrons down a chain from glucose or whatever organic molecule we're talking about, eventually down to oxygen. Oxygen is kind of like our light at the end of the tunnel, drawing electrons toward it. Like supposedly the old stories of a mermaid calling, a siren calling ships onto the rocky shore to the sailor's death. Oxygen is our siren that is calling these electrons, come to me, come to me. And they follow this long chain to get down to the oxygen. So in cellular respiration, glucose and other organic molecules are broken down in a series of steps. We have to have something to carry these electrons. So these electrons are carried from the organic compounds, usually by a coenzyme. Remember, a coenzyme is another organic material or other type of enzyme helper. So this enzyme helper, this coenzyme, is NAD+. NAD plus is very versatile as an electron carrier. So consider NAD plus kind of like the bucket that the electrons are carried in, or the vehicle that the electrons are carried in as they move down these steps toward oxygen, or down this pathway toward oxygen. As an electron acceptor, NAD plus is an oxidizing agent during cellular respiration. So when NAD plus accepts electrons, it is oxidized. It is an oxidizing agent. Whatever it took those electrons from, it's making it more positive. So it is an oxidizing agent to whatever it's taking the electrons from. When NAD plus takes on an electron, then it becomes something called NADH. So it has taken on a negative, it's taken on a hydrogen, and so now it's called NADH. That is the reduced form of NAD plus. Remember, reduced means it is more negative. It's lost some of its positive. So here you can see it's lost its positive. So when it takes on an electron, it becomes what we call NADH. And that represents stored energy. So NADH is in a state of basically temporary stored energy. It's like a temporary battery that then we can tap that NADH and release that energy to create our ATP. So the overall big idea here is that we don't want to release all that energy at one time in a massive explosion. We want controlled small steps, releasing energy along the way. We're going to do that by moving electrons down a chain toward oxygen. We're going to use NAD plus as our enzyme to carry it. When NAD plus carries that electron or accepts that electron, it's functioning as an oxidizing agent. It's oxidizing the glucose or whatever material it took the electron from. It becomes what we call NADH. And that NADH can be thought of kind of as a temporary battery storing energy so that we can use it later to create ATP. So to kind of summarize, NAD plus is the coenzyme, is the electron carrier that transports these electrons kind of through this whole pathway. When they pick up electrons, the material becomes what we call NADH. So NADH then passes those electrons down what we call the electron transport chain. And again, this is kind of like a series of steps, moving down toward oxygen, releasing small amounts of energy along the way instead of just one massive explosive release of energy. So oxygen pulls those electrons down the chain kind of in an energy-yielding tumble down a series of steps, and that energy that's yielded is used to regenerate ATP. So here's a quick little model. We have two ways of doing this, right? We could do it all in one explosive step, an uncontrolled reaction, where we have the hydrogen atoms and oxygen, and all this energy is released in one giant explosion of heat and light and energy. Or the way that it's done in cellular respiration using hydrogen atoms that come from the food molecules that we eat and using NADH as a carrier, it goes down what we call an electron transport chain, and small amounts of energy are released all along the chain. That energy is used to create ATP. Finally, we end up with oxygen at the bottom. Oxygen is the electron, the final electron pull, I guess you could call it, kind of the magnet, sort of, if you want to think about it that way, that's pulling electrons down toward it. And that hydrogen, those protons, are connected with the oxygen at the very end, and water is the end result. Now, very unfortunately, like I said, this requires a number of steps, so it can get pretty complicated pretty quickly. So we're going to take a quick look at a preview here. We're going to break it into 3 steps. So getting energy from the food that you eat, we're going to use glucose as our model, has 3 basic stages. And I don't expect you to memorize every detail of all of these stages, but we do need to know the basics, and we need to think big picture. Keep in mind, what is the purpose here? What exactly is happening? How could we restate this or use an analogy so that we understand? Otherwise, you get very bogged down in the chemistry and the various formulas, and it gets very hard to remember exactly what it is that we're even talking about. So we're going to look at these 3 stages. Here we have glycolysis. Glycolysis is breaking glucose basically breaking glucose in half, and we're going to create what's called pyruvate from that. Then we're going to move to what's called the citric acid cycle, or also called the Krebs cycle. We'll talk about that. That finishes the breakdown of glucose. And then we're going to go into the last step, which is oxidative phosphorylation. Oxidative phosphorylation is the final step, and it's also the step that releases most of the energy and creates most of the ATP. And this is where we have the electron transport chain and the final process. So these are the three sections or stages: glycolysis, breaking glucose down; then citric acid cycle, which is finishing the breakdown; and then oxidative phosphorylation, which is getting out most of the energy. So again, we don't need to know every detail that you're about to see, but at least kind of keep in mind the basic overview and try to push in as much detail as you can. Because this is a complicated process, we're going to try to kind of color code things as we go. So glycolysis, which is the first step, the breakdown of glucose into pyruvate, that's going to be kind of this teal color, and it's the same color they use in your textbook. The second part, which is a citric acid cycle, is going to be kind of this salmon sort of, well, I guess salmon is the best way to describe it, color. And that's breaking down the pyruvate and completing the breakdown of the glucose. And then the purple sort of color is the oxidative phosphorylation. That's the electron transport, and we're going to find out something called chemiosmosis, and that's going to be this violet or purple colored. So the three stages we'll kind of keep in these three colors: teal, salmon, and violet. So again, we're going to use pictures because this is just a real basic overview. We're not going to get into the detail just yet. We'll do that in just a moment, but just a real basic overview. So the first step is glycolysis. So glycolysis is the breakdown of glucose into two molecules that we call pyruvate. And this occurs in the cytosol outside of the mitochondria. So most of the process we're going to look at occurs in the mitochondria, but this first step, called glycolysis, happens outside of the mitochondria in the cytosol. From this first step, we end up with a very small amount of ATP, and we also end up with electrons being carried by NADH. Those electrons are going to move into the mitochondria and go on to the next stage or path. After glycolysis, we move into the mitochondria, and we have pyruvate oxidation. So the pyruvate that we created from the glucose is further changed and becomes something called acetyl-CoA, acetyl coenzyme A. That goes into what we call the citric acid cycle. From the citric acid cycle, which we will find out why it's called a cycle, it produces, again, a small amount of ATP, and then further electrons are carried by two different electron carriers, again, NADH, which is the one that we've already seen, and another one called FADH2. So these two are, again, like temporary batteries. So we've made some NADH over here, kind of like a temporary battery storing energy, and we've made some more temporary batteries here, NADH, FADH2, again, storing and holding energy until our final step. Our final step in the violet here is oxidative phosphorylation, the electron transport chain, and something called chemiosmosis. All of those temporary batteries that we kind of created, the NADH from glycolysis, the NADH and the FADH2 from the citric acid cycle, all of that goes into this final step. Here we have an electron transport chain that we'll talk about, where the electrons finally get to their end point, which is combining with oxygen. And then we also have a huge release of ATP. Oxidative phosphorylation releases the largest amount of ATP. So we had small amount of ATP created here, small amount here, and then the vast majority gets created in this last step. That last step is called oxidative phosphorylation, and we'll talk about why it's called that. Because it's powered by redox reactions. Remember, oxidative means, or if something is oxidized, it means it gave up its electrons. So this whole process at the end is a process of different materials giving up and accepting electrons and moving those electrons down a pathway toward oxygen. And again, you can kind of think of oxygen as the final magnet that is kind of pulling these electrons from higher energy states down through various proteins and materials that we're going to talk about to lower energy states, releasing energy along the way. So we're moving hydrogen atoms, which are a proton and its electron, and we're moving those electrons down a chain, releasing energy along the way. I highly recommend go to the file folder that has Chapter 9 notes and materials. Look for the BioFlix movie about cellular respiration. I think it's very well done, and it kind of helps you understand what's going on because you can visually see it. What I like to do is kind of go over the notes or watch the movie and then go over the notes again. So if you're like me, I go and watch the video perhaps first, or I go and watch the crash course video on YouTube first. So it kind of gives me a basic understanding of what I'm talking about. And then we go back to the notes. I usually go and look at detail in the notes, and then later on I'll go back and watch the video again or the crash course again, because then I've learned more and I can better understand and kind of get the idea that they were talking about. So we're about to wrap up this particular vodcast. Let's just mention again that we have those series of 3 steps. That third step, oxidative phosphorylation, that's where the vast majority of the ATP is made, about 90%. I did say that we have smaller amounts of ATP made in the first 2 steps, glycolysis and the citric acid cycle. That's a process we're going to look at called substrate level phosphorylation. Remember, substrate is a material that connects with an enzyme to get a particular chemical reaction done. So that's involving some enzymes, and there are some reactions that release or help create ATP there, but very small amounts in those first 2 steps. For every molecule of glucose, that's the model that we're using, it eventually is degraded, and the byproducts are carbon dioxide and water. For every molecule of glucose, the cell can make up to 32 molecules of ATP. So 1 molecule of glucose can end up with a final yield of up to 32 molecules of ATP. It doesn't always equal 32 because there are some different pathways that can be followed, but that's the max. So 1 molecule of glucose can get you 32 molecules of ATP. Most of those are created in that last step. Just one last model here, and then we'll take a break. So we said in the 3 steps, we have the creation of ATP. The first 2 steps are very small amounts of ATP. The last step creates a large amount of ATP. The last step we called oxidative phosphorylation because it's part of a redox reaction we're going to look at. The first 2 steps, creating ATP there, is something called substrate level phosphorylation. And this is using an enzyme. And then we have a substrate, which has various phosphorus groups attached to it. And then we have a molecule called ADP. We're going to find out that ADP and ATP are closely related. This is adenine diphosphate. And if you add a phosphate, you create adenine triphosphate. So substrate level phosphorylation is a process where an enzyme combines a substrate and ADP molecule in such a way that it moves a phosphorus group from the substrate to the ADP, and we end up with some byproduct and an ATP molecule. So some ATP is created in this direct fashion like this, using an enzyme and a substrate. That's why it's called substrate level phosphorylation. This occurs in the first 2 steps, in the glycolysis and the citric acid cycle part. This is not the same process that occurs in the end step that creates the large amount of ATP, but we'll look at that in just a second. Let's take a break. I know this is a big, heavy topic. We're going to take bite-sized steps, and we'll move on to concept 9.2.